Hello students, welcome to KENE'S Educare, a group of KENE solutions. This is part 11 of lesson The Fundamental Unit of Life, and this is going to be the last part for the lesson as we are just left with mitosis, meiosis and the exercise question which has been left, right? So we'll be doing that, and then after that we will be doing the exercise questions which we have. So this is basically going to be the last part for the lesson. So I want you all to please have a quick revision of whatever we have studied so far in the lesson, everyone, because here, once we are done with it, you should be able to know what exactly we have studied so far, what is exactly there in the lesson, everything. Okay. So I hope every one of you have started revising it as well. I hope everyone is revising from your book, like whatever notes you have taken so far. So we started the lesson by discussing about the cell theory, about Robert Hooke, right? So I want each one of you to please go through it quickly, go through the slide quickly. After the cell theory, we discussed about cell. So I just want each one of you to please go through this part as well. After this, we discussed on like what are living organisms made up of, right? So I just want each one of you to please go through this quickly. After this activity, we discussed on like what exactly you can see under a compound microscope when you observe the onion peel, right? So please go through this slide as well, everyone. After this, we discussed on activity 5.2. So I just want each one of you to please go through the activity like what is the observation, procedure and the conclusion that you can come up with. After this, like here we have the conclusion to the activity that we were doing, and you can see like the various cells which differ in shape and size have been shown on screen, right? So I just want you all to go through this slide as well. After this, we discussed on division of labor, so I just want y'all to please go through this as well. After this, we discussed on like what is the cell made up of. So what is the structural organization of the cell? And I want you all to please go through it quickly. After this, we discussed on like as we were discussing about the components. Plasma membrane is one such component. So I just want you all to please go through it quickly. After that, we discussed about the diffusion part, so I want every one of y'all to please go through it quickly. After this, we discussed on the three different types of solutions that we have. That is hypertonic, hypertonic, and isotonic solution. So I just want you all to please go through it quickly. After this, like this other drawings that you need to draw, and then we started on activity 5.3. So please go through it, everyone. Then we discussed on this activity 5.4. So I just want you all to please go through it. Then we discussed on endocytosis. So I want you all to please go through it quickly. Then we discussed about cell wall in detail, and I hope everyone is going through it quickly. Then we discussed on this activity 5.6. I want each one of you to please go through it quickly. After this, like this is how the activity is going to look like once you perform it. Then we discussed on nucleus in detail. So I want you all to please go through it quickly. I hope, see, you just don't have to go through the, you can say, written part, okay, the notes. You also have to practice the diagram, because this is a biology lesson, and when it comes to biology, you drawing the diagram is very, very important, okay? After this nucleus, we discussed more about DNA and all, right? DNA, chromatin, chromosome, prokaryotes, eukaryotes, everything. So everyone, please go through it quickly. After this, we discussed on prokaryotic cell in detail. So I want you all to please go through it quickly. After this, we discussed about cytoplasm in detail, and I want every one of you to please go through it quickly, even the structure that has been shown. Okay. Then we discussed more about the cell organelles, where the first organelle that we have discussed was endoplasmic reticulum, where we have studied there are two different types of endoplasmic reticulum that we have, right? One is smooth endoplasmic reticulum and the other one is rough endoplasmic reticulum. After this, here we have the endoplasmic reticulum. So please go through the functions properly. Then we discussed on the difference between the plant cell and the animal cell, and I want you all to please go through it. Not only the differences, we have even discussed on the similarities between plant and animal cell, right? After this, we discussed on Golgi apparatus, and then we discussed on like who exactly discovered Golgi apparatus. So it was Camillo Golgi who did that, right? Then we discussed on Camillo Golgi, like who exactly he was and how exactly he came up with it, right? After this, we discussed about lysosomes, and we discussed why lysosomes are called as the suicide bags, right? So I want you all to please go through it quickly. I hope you all are going through the diagram as well. After lysosomes, we discussed on mitochondria, like what exactly mitochondria is. Even there is like cristae, matrix, inner membrane, outer membrane. Okay, everything is given in the structure above. So when you are going through all the points, you need to go through the structure as well. I hope everyone is going through it properly. After this mitochondria, we discussed on plastids as well, like what exactly plastids are, and always remember that plastids are present only in plant cell, they are not present in animal cell. Okay, and you should be even knowing the functions of it. Then after this plastids, we discussed on vacuole, like what exactly vacuoles are. And always remember that plant cells have bigger vacuole and animal cells have smaller vacuoles. And the main store, like function of vacuole is it act as a storage sac. Okay. I hope everyone is going through the functions of vacuoles that has been given on screen. After this vacuoles, we had some more important points of vacuoles which have been given. So I just want each one of you to please go through this as well, because after this we will be discussing about cell division, that is about mitosis and meiosis and detail. Okay. After this, now we have cell division to discuss. So see, now you can see what exactly is happening in mitosis and meiosis. The diagram is clear, right? See, the cells are formed in organism, and the reason of forming those cells are like because we how we are growing, because the cells are getting formed. The torn out cell, the dead cells, injured cells, they are getting replaced by new cells which have been formed in the body, right? So new cells are formed in organism in order to grow, to replace the old one, then to replace old, dead, and injured cells and form gametes which are required for reproduction. Now, for reproduction, gametes play a very important role, and for that, the cells help, like mitosis plays a very important role. Okay, so the process by which new cell is made is called cell division. What do we call it as? Cell division. The process by which new cell is new cells are made is called cell division. And there are two types of cell division. One is mitosis and the other one is meiosis. Okay, so the process of cell division by which most of the cells divide for growth. Okay, if the cell is getting divided for growth, then we call that process of cell division as mitosis. And in mitosis, each cell called mother cell is going to divide and is going to form two identical daughter cell. How many? Two identical daughter cells will be formed. So when a mother cell is getting divided, it forms two identical daughter cell. We know that kind of cell division as mitosis. And the daughter cell which has been formed, it will have the same number of chromosome as the mother cell. Okay, and mitosis is mostly going to help in growth and repair of tissue in organism. Where it is going to help? It is going to help in growth and repair of the tissues. Okay, so first para is giving us more information about mitosis. Now the second paragraph that we have is giving us information about meiosis. So see, mitosis, what was happening with respect to growth and repair of the tissues, right? Meiosis happens if there are certain specific cells of reproductive organ or tissue in plant and animal cell, and if those cells are going to divide to form a gamete, then there, for helping the reproductive organ and tissues to divide, meiosis plays an important role. Okay, so which after fertilization give rise to offspring, and they divide by a process called as meiosis. How do they divide? By process called meiosis. And in meiosis we have two types, like two consecutive divisions which happen. In mitosis we have only one division, whereas in meiosis there are two consecutive process that takes place in meiosis. Okay, so when a cell divides by meiosis, it produces four cells instead of just two, because in mitosis only two daughter cells are formed, but in meiosis four daughter cells have been formed. Okay, so the new cells only have half the number of chromosome. Now in mitosis only two cells were formed, but they had the same number of chromosome as the parent. But over here in meiosis, the new cells, the four cells which are been formed, they're going to have half the number of chromosomes uh, than that of the mother cell. So can you think as to why the chromosome numbers have reduced to half in mere daughter cells? What do you think? So like I hope you all have like got the difference that exists between mitosis and meiosis, right? So I just want you all to draw both the diagrams of mitosis and meiosis and write all the points in your book as well. I hope everyone is taking proper notes on mitosis and meiosis both. I hope everyone is drawing the diagram and is doing it properly, taking note of it properly. Because after this we have the summary discussion and then we have few exercise questions to discuss. We'll be doing that and then like the lesson will be over. So I want you all to please be quick with it. Now after this we have summary discussion over here. So I want you all to please read each and every question, like every point which has been given. And if you find that you have any doubt in any of them, please you can like feel comfortable to ask it. I hope everyone is reading all the points which have been mentioned in the summary part, as that is very important for you all to know it. Okay, and like you should be able to understand each and every point given in the summary, because that is what is going to make you confident enough that you know the concepts as well. After this, now we have the exercise questions to discuss. Okay, the first question given over here is make a comparison and write down ways in which plant cells are different from the animal cell, right? So we have already discussed on this part in last to last lesson, right? If you remember. So tell me now what are the differences that you can write and what are the similarities that you can write between the plant cell and the animal cell. It's very easy, right? See, what you can write is about the cell wall. You know, cell wall is present in plants, whereas it is absent in animal cell. You can write that one differentiation. You can write about the nucleus, that in plant cell, the nucleus lies on one side of the cell, whereas in animals, it is present in the center of the cell. Then lysosomes are rarely present in plant cell, but in animal cell, lysosomes are always present, right? Then we studied one more thing about plastids, that plastids are present in plant cell and they are absent in animal cell. Then we even discussed one more part about plant and animal cell is about the vacuole we discussed. Yeah, plants will have large vacuole whereas animals will have small vacuole. So these are some of the differences that you can write. Okay. Then how is prokaryotic cell different from eukaryotic cell? So you know the size of the prokaryotic cell is generally like 1 to 10 micrometer, whereas of eukaryotic cell the size is from 5 to 100 micrometer. So for prokaryotic it is 1 to 10 micrometer, for eukaryotic it is 5 to 100 micrometer. Okay, then prokaryotic cell will have a well defined nucleus, whereas eukaryotic cell do not have well defined nucleus. Then there is only one single chromosome present in prokaryotic cell, sorry, single chromosome, whereas in eukaryotic cell there will be more than one chromosome which will be present. Okay, and then in prokaryotic cell the membrane bound cell organelles will be absent because the nucleus is not well developed. Lysosomes, ribosomes, endoplasmic reticulum, these are the membrane bound cell organelle and they will be present in prokaryotic cell, whereas in eukaryotic cell the membrane bound cell organelles will be present. Okay, so these differences you need to mention. Then what would happen if the plasma membrane ruptures or break down? So see, if the plasma membrane ruptures or break down, then molecule of some substance will freely move in and out of the cell. Now if you remember, plasma membrane is a selectively permeable membrane and it acts as a mechanical barrier, right? So the exchange of material from its surrounding through osmosis or diffusion in the cell won't take place. What will you write? The exchange of material from its surrounding through osmosis or diffusion in a cell won't take place, and consequently the cell will die due to the disappearance of protoplasmic material. Okay, so protoplasm, like plasma membrane, is very important for the cell. Okay, so it should not get ruptured or it should not break down. Then fourth question that we have is what would happen to the life of a cell if there was no Golgi apparatus? Now we know that Golgi apparatus, it consists of stacks of membrane bound vesicles, and the function of this membrane bound vesicle is storage of substance, packing of substance, manufacturing of substance, right? So storage, first of all manufacturing it, packaging it, and lastly storing it. These are the three functions of the membrane bound vesicles which are present in Golgi apparatus, right? So without the Golgi apparatus the cells will disable from packing and dispatching the materials that are produced by the cell, and the Golgi apparatus is also involved in formations of cell, right? So if there is no Golgi apparatus, cells will not be produced. Got it, everyone? So this is what you need to elaborate. Then which organelle is known as the powerhouse of the cell and why? So we know the historic statement that mitochondria are known as the powerhouse of the cell, and it is because it releases the energy required for different activities of life. Whatever activities that we are performing, we need energy for that, and that is being released by mitochondria in the form of ATP. ATP stands for adenosine triphosphate, right? So this ATP is essential for numerous chemical activities, and that is why very often we also call this ATP as the powerhouse of the cell. What do we also call it as the powerhouse of the cell. Okay, then after that, where do the lipids and proteins constituting the cell membrane get synthesized? So lipids and proteins, they are synthesized in the endoplasmic reticulum, that is ER, right? Then the next question that we have is, how does an amoeba obtain its food? So we know that amoeba obtain its food through the process of endocytosis, right? Because the cell membrane is flexible enough, amoeba is going to engulf the food particle, forming a food vacuole, gilding it, and it is assisted by the pseudopodia. Okay, and over here, amoeba is going to secrete some digestive enzyme which is going to carry out digestion of the engulfed particle once the food has been trapped in. I hope everyone is getting it. I hope everyone is writing it. After this, like, you have to explain about the endocytosis process in detail. Then after that, we have the next question as, what is osmosis? So the process of movement of water molecule from a region of higher concentration to a region of lower concentration, and that is through a semi-permeable membrane. We know that process as osmosis. How will you define osmosis? The process of movement of water molecule from a region of higher concentration to a region of lower concentration through a semi-permeable membrane is known as osmosis. So you have to write the definition of it. Now here we have the ninth question as, carry out the following osmosis experiment, where you need to take 4 peeled potato halves and scoop each one out to make potato cups. Now one of these potato cups should be made from a boiled potato, and put each potato cup in through containing water. Okay, and now keep cup A empty. Put 1 teaspoon sugar in cup B. Put 1 teaspoon salt in cup C. Put 1 teaspoon sugar in boiled potato, cup D, and keep this for 2 hours. And then observe the 4 potato cups and answer the following. So you have to explain why water gathers in the hollowed portion of B and C. So what you can write over here is, water accumulates in the hollowed portion of B and C as a difference in the water concentration is observed, and therefore endosmosis occurs as the cells act as a semi-permeable membrane. Okay, then why is potato A necessary for this experiment? Because we need to compare the different scenarios, right? We need to compare cup B, C and D as well. So for comparing them, potato A plays a very essential role in the experiment. Okay. Then third one, explain why water does not gather in the hollowed out portions of A and D. So cup in A does not show any change in the water flow concentration for osmosis to occur, right? So because why is it so? Because it requires concentration to be higher than the other. And the cells in cup D, they are dead. Thus there is no existence of semi-permeable membrane for water flow. And subsequently what happens? Consequently what will happen? Osmosis does not occur. I hope everyone understood this experiment and the and how to answer the questions. I hope everyone has understood this, like what exactly happened over here. Now, after this, the 10th question that we have is, which type of cell division is required for growth and repair of body, and which type is involved in the formation of gametes? So we know there are two types of cell division, right? One is mitosis and the other one is meiosis. And we know that mitosis is required, it is involved in the growth and repair of the body, whereas meiosis is a type of cell division which results in the formation of gametes. What does it results into? It results in the formation of gamete, and mitosis results in the like is required for growth and repair of the body. Okay. So this is how we are ending up with the lesson fundamental unit of life, and I hope that whatever we studied and discussed today in this part for the lesson, like exercise, mitosis, meiosis, and about the cell organelles, each and everything that we have studied so far in the lesson, every one of you are very much clear with it, right? So here we complete with the whole lesson along with the summary, along with the exercise question, each and everything. Now I've told you all the answers orally, so you have one homework that you need to write the answers on your own. Okay, you don't have to take the help of Google, don't have to take the help of your textbook. Whatever you know, just write it and then you can just cross verify it by checking it through your textbook or the source that you have. Okay. So here I'll end up with the lesson fundamental unit of life, and I'll start with the next lesson in the next part. Thank you everyone.